The Golgi apparatus.
The Golgi apparatus serves as a processing center for the exportation of proteins, lipids, and other large molecules to their final destinations outside of the cell.
Golgi apparatus or Golgi complex is a membrane-bound organelle which is made of eukaryotic cells and is made of flattened stack pouches that are called cisternae. The Golgi apparatus is responsible for delivering and transporting and modifying proteins and lipids into vesicles, which are made to deliver the proteins and lipids into targeted destinations. The apparatus has three primary compartments, known as CIS, which are cisternae nearest to the endoplasmic reticulum, media, which is central layers of cisternae, and trans, which is cisternae farthest from the endoplasmic reticulum. And now I'm going to talk about the two most important, which is the CIS Golgi network and the trans Golgi network.
The trans Golgi network functions in the processing and sorting of glycoproteins and glycolipids at the interface of the biosynthetic and endosomal pathways. The generation and maintenance of apical and basolateral membranes rely on sorting events that occur in the TGN.
The cis Golgi network and the trans Golgi network are used to sort the proteins that are received in the organelle and furthermore to be released in the other parts of the organelle.
The cis Golgi network is an extensive tubulovesicular network bound to the cis face of the Golgi stack and which function is to receive process the biosynthetic output from the ER.
Each cisternia or region of the Golgi apparatus contains different protein modification enzymes, which means that those enzymes have different work in the cell, and the Golgi enzyme catalyzes the addition or of removal of sugars from cargo proteins, which is the glucosylation, the addition of sulfate groups, which is sulfation, and the addition of phosphate groups, phosphorylation, that's right. And those cargo proteins are modified by enzymes which are called resident enzymes, located within the cisternia of the cell. And the enzymes sequentially add the appropriate modifications to each of those cargo proteins. Some Golgi-mediated modifications act as signals to direct the proteins to their final destinations within the cells, including the lysosomes and the nucleus of the cell. The way in which proteins and lipids move from the cis phase to the trans phase is of some debate, which means that there are two theories that scientists developed how proteins and lipids move from the cis phase into the trans phase. The first one is the vesicular transport model, which means that identified vesicles but often fuse to the cisternae membranes, thus moving the molecules from one cisternia to the next one. Furthermore, it means that they're not doing the whole work, unlike the second theory, which says that which is the cisternal maturation model, which depicts the Golgi apparatus as far more dynamic organelle than the first one says, and states that it does all the work, unlike the first one, which says that only they transport it. But in the second one, says that the cisternal maturation model indicates that the cisternae move forward and mature into transcisternae, with new cisternae forming in focus of vesicles in the cis phase. In this model, vesicles are formed but are only used to transport molecules back from the endoplasmic reticulum, unlike the first one, which says that they move used to transport molecules back to the endoplasmic reticulum, which is the ER. The Golgi apparatus processes proteins and lipids made by the endoplasmic reticulum, which is known as the ER of the cell, and it does that before sending them out of the cell. And the proteins that enter the Golgi apparatus on the side facing the ER, which means that it will be on the cis side, which is right here. Exit on the opposite side, as you can see. Proteins must make their way through the stack of cisternae and along the way become modified and packaged for the transport and be able to exit the cell. And the Golgi apparatus cisternæ vary in number, as I already said. In different cells there will be a lot more, but there will be very small, but the others, maybe in animal cells, there will be in few amounts, but they will be very large. So this is the difference in the Golgi apparatuses between the different kind of cells.
So now I'm going to be talking about
a ribosome is a cell organelle. It functions as a micro machine for making proteins. And ribosomes are composed of special proteins and nucleic acids. The translation of information and the linking of amino acids are the heart of the protein production process.
The word ribosome is made from taking ribo from ribonucleic acid and adding it to soma, which is the Latin word for body. Ribosomes are organelles composed from ribosome proteins and ribonucleic acids. Ribosome is basically a very complicated but elegant micro machine for producing proteins. Each complete ribosome is constructed from 2 subunits. Ribosomes are found in prokaryotic and eukaryotic cells, in mitochondria, in chloroplasts and bacteria. Those found in prokaryotes are generally smaller than those in eukaryotes. Ribosomes in mitochondria and chloroplasts are similar in size to the ones in bacteria. Ribosomes are found free in the cytoplasm or bound to the endoplasmic reticulum or the ER to form rough ER. Several ribosomes can be attached to the same mRNA strand. The structure is called polysome. Ribosomes have only a temporary existence. When they have synthesized a polypeptide, the 2 subunits separate and are either reused or broken up. Ribosomes are cellular organelles that carry out protein synthesis through a process called translation. They're found in both prokaryotes and eukaryotes. These molecular machines are responsible for accurately translating the linear genetic code via the messenger RNA into a linear sequence of amino acids to produce a protein. All cells contain ribosomes because this growth requires the continued synthesis of new proteins. Ribosomes can exist in great numbers, ranging from thousands in a bacterial cell to hundreds of thousands in some human cells and hundreds of million thousands in frog ovum. Ribosomes are also found in mitochondria and chloroplasts. The ribosome is a large ribonucleoprotein or RNA protein complex roughly 20 to 30 nanometers in diameter. It is formed from 2 unequally sized subunits referred to as large subunit and small subunit. The 2 subunits of the ribosome must join together to become active in protein synthesis, as we can see. To summarize, we can say that nearly all proteins required by cells are synthesized by ribosomes. Their ribosomes are found free in the cell cytoplasm and are also attached to rough ER. Ribosomes translate information encoded in messenger ribonucleic acid, and they link together specific amino acids to form polypeptides, and they export these to the cytoplasm. Also, a mammalian cell has as many as 10 million ribosomes. However, each ribosome has only a temporary existence, and ribosomes receive information from the cell nucleus and construction materials from the cytoplasm.